Hey all, welcome back. This is me, Monica Sharma, and welcome to MS Bio Academy. And today we are going to talk about a very interesting topic, that is about how to calculate the number of chromosomes and how we can determine that what is the number of chromatids or the DNA content is present in the different phases of mitosis. Now, why I have taken this topic? Because I have found that most of the students are getting confused with this particular topic and they are not able to find it out that what is chromosome number, what is the amount of chromatid is there in different phases of mitosis. So let's now discuss this and clear your doubts, each and everything, and let's make it very simple to understand and memorize that how many number of chromosomes are present, how many number of chromatids are present, what is the DNA content is present in different phases. So let's start it. So there are some basic rules you need to memorize before finding out that what is the number of chromosome or chromatid is there in different phases, right? So what are those rules? The very important rule is this, that the number of chromosomes, how are you going to find it out that what is the number of chromosomes present? Just count the number of centromere. So whatever number of centromere will be there in the image, that will be your number of chromosomes. So if one centromere you see, there will be a one chromosome, right? If two centromeres you see, there will be a two chromosomes, like that way. For example, I'll give you one example. Let's suppose this is your this is the centromere with which the chromosomes arms are attached, right? So you know that is a P arm and the Q arm, both are attached, and this is a centromere. So you can see here there is a one centromere is seen. So that means this is the one chromosome only, right? Now see this one. What about this one? This is again centromere. These are the arms. So what do you see? Again one centromere. So that means this is also together a one chromosome only, right? Now if I give you like this one, let's suppose this one. Okay. Now what do you see here? There are two centromeres you can see. That means this is one chromosome, this is one chromosome. That means two chromosomes are present, right? Similarly, if I give you like this one, okay, so here also you can see there is a two centromeres are present. One is this, one is this. That means these two are the two chromosomes, right? This is one chromosome, this is another chromosome. So two chromosomes again. Now talking about the chromatids. So now you understood how to find it out the number of chromosomes. Now let's find out the number of chromatids. Now number of chromatids. Chromatids, first of all, what is it? Chromatid is nothing but the copies of the chromosomes. Okay, so the copy of the chromosome, when the chromosome, like you know that in the cell cycle there are, what are the phases are there? There is a G1 phase is there, then S phase is there, then G2 phase is there, and then M phase is there, right? Like this way it goes always. This is your cell cycle, right? So in the S phase we know that the DNA gets replicated, right? So if the chromosome is like this way, and during the S phase it will become replicated, means it will form its own copy, right? So one more copy is being formed here. So now this second copy which is now being formed, this is nothing but the sister chromatid. So these two are the sister chromatid. This is one sister chromatid, this is second sister chromatid, right? So there are two sister chromatids are there. So I can say here that this is the one chromosome and one chromatid I can see, right? And here again I can see one chromosome since one centromere is present. But what about the chromatids? There are two sister chromatids are there, right? So I'll write SC for sister chromatids, two sister chromatids. Again here we have seen two centromeres are present, so two chromosomes. But what about the chromatids? There is two here and two here. So that means there are total four sister chromatids. Similarly, see here since two centromeres, so two chromosomes. But what about the chromatids? This is one sister chromatid, this is second sister chromatid. So that means two sister chromatids are there. So easy, this concept is clear. So wherever you will see such kind of images, just find it out the number of sister chromatids and the chromosomes like this way. So this is clear. Now what is the second rule says? Second rule says that the amount of the DNA content is equals to the number of chromatid. Now if somebody will ask you what will be the amount of DNA content will be present, and do remember that DNA content we always represent with the C letter. Okay, so normally you will be asked that what is the DNA content during, let's suppose, in the gametes. Okay, so if I talk about the gametes, you know that the chromosome number, we represent it with the letter N, right? So in the gametes, the chromosome number, that is N, is equals to what? If I talk about the human beings, it is 23, right? We have 23 pairs of chromosome. In haploid set, it is 23 chromosomes, right? In 2n set, it will be 2 into 23, that is 46 chromosomes, right? So if I talk about in the gametes, there is a N is equals to 23. And what about the C content? The C content will be 1C only. Okay, so 1C, why? Because here the DNA is not being replicated, it is present in the single copy, so that will be the 1C only. All right, since chromosomes is 23, so 1C content. But let's suppose one more case. Now this is one gamete, this is another gamete. Okay, so this has N and 1N, and this is 1C. This is again 1N and 1C. When they undergo fertilization, what they form? They form the zygote, right? So now here what will be zygote will become 2N, and now the DNA content will also become 2C, right? So if this is your, let's suppose this is your chromosomes, okay, so now here it will become two copies of these chromosomes, like this way. So 2N and 2C, the DNA content has doubled now, right? Similarly, when this cell will enter into the G1 phase, now what will be the DNA content? G1 phase is nothing but the growth phase, so there will be no change in the chromosome number and DNA content, so it will remain 2N and 2C only. But what about the S phase? S phase, as I have told you, it will be DNA replication will take place, so that means here the chromosome number will remain same, 2N only, but the DNA content, since the DNA gets replicated, the DNA content will become from 2C, it will be duplicated to 4C. Okay, so if here the chromosomes are like this way, let's suppose this is small, this is big, this is small, this is big, here it will be duplicated like this way. Okay, so now the DNA content is doubled, so it has become 4C, but the chromosome number will remain the same, 1, 2, 3, 4, means diploid site will only be there. So that is how you can find it out, that how the DNA content and the chromosome number will keep on changing. Similarly, let's go for the G2 phase. In the G2 phase also it will remain the same, because in G2 phase nothing happens to the DNA content or chromosome number, it is just the growth phase, right? G2 and G1 are just the growth phase, so there is no change in the chromosome number, DNA content. Only in the S phase the DNA content gets doubled. Okay, so here also, since the S phase the DNA content was 4C and chromosome number was 2N, so here also 2N and 4C will remain. Now what about the mitosis phase? When it enters the mitosis, it will form the two daughter cells, right? So here the two daughter cells will get what? 2N, 2N each, right? In the mitosis, the diploid cell will be restored in the two daughter cells also, correct? We know this. It will become diploid only. So 2N, 2N will remain, but what about the DNA content? The DNA content will again be restored to the 2C, right? So 2C, 2C, one copy each will be gone to the daughter cells. So that's why this is become 4C here, because it needs to be equally distributed in the two daughter cells, right? So that is how you can find it out, the DNA content, and you can find it out the number of chromatids and the number of chromosomes. Now let's discuss in more details with respect to the chromatids and the chromosome number here. Now before replication, or before you can say mitosis, this is the condition. Okay, now this is the condition when the replication has occurred. Okay, after replication, you can say after replication. Now again, just after mitosis, this will be the condition. Okay, so this is after mitosis. So what you can say here, the chromosome number is 1. So if I write it down N here and there, let's suppose here I write chromatid number, and this is your chromosome number, chromosome number. So let's write it down. So here chromosome number 1 centromere, so 1 chromosome, 1 centromere, so 1 chromosome, 1 centromere, so 1 chromosome. What about the chromatid? Chromatid is 1 only. Here the chromatid is 2. Now here the chromatid is again 1. So you can see the transition that before mitosis, after mitosis, the chromosome number remains same, but the chromatid number changes after replication, but it is restored back after the mitosis. Alright, so this is the basic step. Now let's talk about it in different phases of mitosis. We'll talk about the prophase, metaphase, anaphase and telophase. So this is your prophase, this is your metaphase where the chromosomes are aligned at the metaphase plate, then the anaphase where the sister chromatids will separate, and this is your telophase, final cell division will take place. And this is further followed by the cytokinesis, right, where the complete cell division take place and the individual 2 daughter cells will be formed. Right, so here you can see these are the homologous chromosomes which are being shown here. So if we talk about here that what is the number of chromosome, let's try it together. So you can see here the 1 2 3 4 centromere, so that means here chromosome number, let's write here chromosome number, and this is your chromatid number. Okay, so chromosome number is here 4. And what about the chromatids? Chromatids you can write 2 plus 2 4, 6, 8, right? So chromatid is 8. Now talking about the metaphase. Metaphase again you can see 4 centromere, that means 4 chromosomes. And what about the chromatid? Chromatids is again the same, what is 8, right? Now in the anaphase, the centromere, these chromosome or the sister chromatids is separate and they move towards opposite poles. So here now this will become 1 chromosome, right? So here the total number of chromosome will be total number of centromeres. So just count it down, 1 2 3 4 5 6 7 8, right? So 8 chromosomes. And what about the chromatids? 1 2 3 4 5 6 7 8. So 8 sister chromatids, 8 chromosomes. Again here also the same way you can see that there is the 8 chromosome and 8 chromatids, right? After cytokinesis, what will occur? After cytokinesis you will get, again here you can see that there are 4 centromeres are present in each cell, so that means 4 chromosomes are there. Okay, in each of these cells, here also 4 chromosomes are there. And what about the chromatids? Chromatids also you can see that 4 chromatids in each of them. So 4 is again restored here. So this is how you can calculate it. Now if I summarize this thing in different phases, how you will calculate for the, let's suppose for the human beings, the number of chromosome is being asked to you in the questions. So how you will say that what is the number of chromosomes, what are the number of sister chromatids. So let's take in this column, we'll take the number of chromosomes, and this is during mitosis only we are talking about. We are not taking the consideration of meiosis here. We'll talk about in the next video in the meiosis, right? So here this is the number of chromatids. So in interphase, during the interphase, what happened was there is a 2n number of chromosome will be present. Diploid cell only undergoes mitosis, right? So 2n. So since we know that n is equals to 23 in humans, so 2n is equals to 46 chromosomes and 46 chromatids only. Now when the S phase has occurs, the replication will take place. So what will happen? The 2n number is still there, so that means chromosome number will remain the same, 46. But what about the DNA content is doubled, so that means each of the 46 chromosome will be now like this form, right? So there will be each chromosomes will contain 2 sister chromatids, right? So that means 46 chromosome will contain how many? 92 chromatids. So there are 92 chromatids will be present, but 46 chromosome will be present. Then metaphase also there will be diploid set only, right? Now here also again the chromosome will be 46 only, because they just arranged in the metaphase plate only, in the metaphase stage nothing will change. So 92 chromatids only. Anaphase stage, what happens? Anaphase stage, there is a chromosome number gets doubled, right? So it becomes 2 multiplied by 2n, means it will become 4n here, right? So that means here 92 chromosomes will be present, right? Remember anaphase, I told you, see this will become separated, right? So it will become from 4 to it becomes 8, right? So here it becomes 92. And what about the chromatids? Chromatids will remain the same, the 92 only, right? And what about the telophase? Telophase is again 4n. Here also 92 chromosome, 92 chromatids. What about the cytokinesis? Cytokinesis, again the cell restores back to its 2n number. So 2n in each daughter cell. So 46 chromosomes, and chromatids also becomes 46 here. So this is the whole summary of the chart. If any question comes related to this, I hope now it is clear to you that how to find it out this thing. It's very easy concept. So just follow these basic rules which we have discussed, that number of chromosome is equal to number of centromeres, and number of chromatid is equals to, it only depends on that if the chromosome become duplicated during the S phase, after the replication, after the DNA replication, then the chromatid number will be 2 for one chromosomes, right? So like this is the one chromosome, this is a duplicated form of the chromosome after replication. So that means there are 2 chromatids, but it is one chromosome. So that is how you can calculate it. So that's it for today. I hope you liked this video. If you have any query related to this topic or any other topic, please do comment down below in the comment section. And thanks for watching, and don't forget to subscribe MS Bio Academy. Bye for now.